Okay, so we covered inoculation, media and stuff. So next, what do we do with inoculated plates? Incubator. We stick them in the box. It maintains the temperature, 37 degrees Celsius. Why do we put them in the incubator? Why don't we just leave them on the table? So first of all, it provides the favorable environment. A lot of bacteria grow better at 37 than they will at the room temperature, something else. What would you choose? The incubator set up at 25, or, well, incubator set up at 70 Fahrenheit, or just table? So it's 70 in the room. So you have a choice. Incubator at 70, or just here. Why? Why do you want incubator? Yes, it's controlled environment, so everything is standard. Why do you want controlled environment?
So that way they can grow and you can see the progress. Like if it's in a controlled environment, it'll grow faster.
I mean, if I set it on 70, they're going to grow slow, but it's still better than to be on the table. And trust me, they will grow on the table okay. Why controlled? Huh? Excellent. Reliable results. We can compare the results. Does that make sense? We eliminate multiple variables. If we want to study something, we can leave one variable. Right? Make sense? If I grew my cells at 37, then you can grow your cells at 37, and we can compare the results. All right? And then we do isolation, we subculture. I'm going to talk more about techniques of subculturing. So the plates that you have seen after your hand wash experiment had multiple species, right? Yellow, white, and stuff. If you want to isolate just one, you have to take that colony, pick it up, pick a little bit of microbes from that colony, like you did for wet mount, and place it either in a liquid medium or in the plate. So what's going to happen to microbes in the liquid medium? Huh? And what, are they going to divide? In the medium? Sure, yeah, they're going to grow, right? So you're going to get a lot of microbes in the liquid medium. But for some reason, sometimes you want to take like a white colony and put it on another plate. Why do you want to do that sometimes? Spread it through another plate, subculture it on another plate. Although you know it's a white colony. Why? Why would you want to do that? Okay, yes, how it grows on its own. In terms of the preparation of the pure culture, why do you want to take something that's apparently pure and put it in another plate? A little bit developing your statement so we know that it's pure. Because we did with students on multiple occasions, somebody has a culture from the environment, okay, the mixed culture, and wants to cultivate, I don't know, yellow. Picks up yellow, spreads on the new plate, and it comes out 2 or 3 species. Yellow just masked 2 others. Does that make sense? So we want to make sure that the colony is homogeneous, that there's one type of microbes in this colony. So don't have any contaminants. Does that make sense? Okay. How do we separate? How do we put them on the plate? There are 3 major techniques: the strict plate, the spread plate, and the pour plate. Basically, what we refer to is when you have a mixture of microbes, you separate them on the plate. So now you can see different colonies. This is an example of a plate when they're not separated well enough. You can tell that there are multiple species, but you cannot pick one colony without touching another. When they are separated well enough, you can clearly see 3 different species. You can see the shape of the colony, the color, everything. How you do that? The strict technique, and I mean, picture's fine, but I want to show it to you, like draw it for you. The only problem is that I don't want to switch to notebook, like a white screen, because it may screw up the computer. So I'm gonna have to do that here. So this is your plate with agar. So what you do, you divide the plate for inoculation like this. It doesn't have to be this 3 parts. It's just easier to show with this 3 parts. Okay, so you divide your plate. You just draw with a Sharpie. Then you pick your colony, whatever you wanna pass, right? Or whatever sample you have, like environmental sample. We're gonna do that with environmental samples, right? You take the cotton swab, you swipe, I don't know, ear. Okay, by the way, you can do it next week. You can swipe your ear, your nose, whatever. I mean, if something requires restroom, go for it. I don't mind. And you inoculate this part. Clear? Then you throw away the cotton swab, pick up the sterile loop. Sterile. Grab a little bit of microbe from here and start to spread it. That makes sense? So you get a little bit of what you've already put on the plate. So essentially you do the transmissibility thing on one plate. Does that make sense? So this way, whatever was here, okay, whatever was here is getting spread through the large surface. Make sense? Next, pick up a little bit from here and spread it this way. So every time between this spreads, you sterilize the loop. So you always touch your microbes with a sterile loop. Got it? This way you kind of thin out the layer of bacteria. You dilute them. You spread the smaller amounts across the large surface. Does that make sense? So every time you have less and less microbial cells, and they're going to form less and less colonies, and colonies will be more separated. Is that clear? You don't have to do the picture. This scheme here shows you how you can do it, in slightly different way with more spread. It's entirely up to you. I just find the dividing part much easier to handle and execute, because you kind of have a guideline. I put my initial sample here, spread it over the second quarter, and spread over the third quarter. Good? We're good? No questions? Move on to the pour plate and spread plate. Pour plate. You have agar in the liquid shape yet. It's warm enough to stay fluid, but it's cold enough. I mean, it's not hot. So microbes are not going to die when they get into it. So collect the sample with a sterile loop and dip this loop into the first tube with agar. You mix it. Then you sterilize the loop and pick the sample from the first, move it to the second. Mix it. Sterilize the loop. Move from the second to the third. You do serial dilutions. right? Every time the number of cells, microbial cells, in the tube is going to be progressively lower. When you pour that agar in the Petri dish, you're going to see the separation here. So you see the number of cells lower, and now you can separate the colonies. Make sense? That's why it's called pour plate. Yes? Say it one more time. Usually we don't use swab. We have to use this. Does that make sense? So if you want to do that, you gotta have the wire loop. You still can swab technically if you to inoculate first tube. Then okay. 3 tubes. Okay. Tube number 1, my index finger. I sterilize the loop. I collect the sample. I inoculate. I shake. I sterilize the loop. I take the sample from the first, move it to second. I shake. I sterilize the loop. I move the sample from the second to third. I shake. I pour. You got your growth. Spread plate. Much simpler. You get the hockey stick. You put your sample on the plate and you spread it using hockey stick all over. That's it. It's not as good for purpose of separation of colonies because you have a ton, they're not going to get separated, but it's really good to prepare so-called bacterial lawn. When you spread them evenly, you may get a plate which is completely covered with colonies so that they almost fuse. Make sense? Okay. Inspection and identification. Inspection. You've done it already, right? Yellow, white, large, small colonies. It's all inspection. You inspect your plate. Did your media change color? Do colonies look different on one plate than on another? Okay? You see, that's inspection. But it doesn't really, I mean, unless you know, look, let's say you walk in microbiology class, you never took microbiology before, I show you the plate, the hand wash, and tell you, describe me the plate. What you gonna tell me? The hand wash, your hand wash plate. What does it have? Well, you don't know that it's bacteria. Circles that are colored, right? That's your inspection. You don't really know yet what it is. So unless you can identify species, you can't be 100% sure. How can you identify species? You can do the microscopy. And in many cases, if you can stain, using gram stain, for example, you can tell apart gram positive, gram negative. You see the shape, and especially when you know the source. Okay, you can say, yeah, these are gram positive cocci that come from the nose. They are hemolytic in the blood agar, and they form large clusters of cells under the microscope. That's Staphylococcus aureus. Because it's Staphylococcus, large clusters of round cells. It's gram positive. and it's hemolytic, which is pretty common for Staph aureus. Often at this point, you can quite convincingly, demonstrate what you're dealing with. And actually, in many cases, it's kind of enough. For purposes of treatment in clinical diagnostics, that's pretty much it. The selection of antibiotics often will be based on whether the microbes are gram-positive or gram-negative, or acid-fast. I can give an example when, I bring a lot of examples from my personal family experience. My son had bone infection. He was like 10, something like that, 9, I don't remember. Anyway, had bone infection. So they took a biopsy of the bone. And when biopsy results came in, the ID doc was treating my son. He looked at the results, and I looked at the results with him. And it's like hugely, but he didn't even look. Results came. He called the lab and said, "Is that myco?" So he was worried that it may be Mycobacterium tuberculosis. Is that myco? No, it's not. It's not tuberculosis. Then I don't care. Then I know what to prescribe. And as you remember, you can do acid-fast stain for quick identification, right? So you can quickly decide. And then if it's not Mycobacterium, he said, we know how to treat. Does that make sense? So often the decision does not require the exact identification of the pathogen, right? But still, we want the diagnosis. We want to know if patient has plague or something less dangerous. So there are different types of tests. Tests can be based on immunological recognition. They can be biochemical when you analyze the products of microbial metabolism. Or they can be genetic when you analyze the genetic makeup of the microorganism. And we're going to talk about those tests. So first is going to be immunological methods. And they, how many of you are familiar with the concept of antibody? Yes? Okay. Almost everybody, and this is good. But I will just remind you a little bit about what antibodies are. Sounds good? So when the large molecule, say protein, that does not belong to ourselves, enters our bodies, so-called antigen, a foreign molecule, when it gets into the human organism or mouse organism or a lot of animals, okay, it stimulates the immune system to produce other molecules that are called antibodies. Antibodies can recognize the antigen, and they are extremely specific, like lock and key. They are unbelievably specific. Is that clear? All immunological methods that we're going to talk about, and it's not an exhaustive list, are based on the concept of recognition. When antibody recognizes antigen. And immunological methods designed in a way that we have antibodies to antigens we're looking for, right? For instance, HIV can be detected using that type of recognition, okay? You can take, in some cases, it's opposite. We look for antibodies, not the antigen. But okay, let's say you have a culture of microbes. You don't know what it is, but you have a suspicion that it can be Streptococcus pyogenes. Okay, so you can take antibodies that recognize very specific M protein of Streptococcus. and then try to detect the complexes that they form. And I will show you how it works. Since antibodies recognize proteins, can recognize proteins, one method is based on detecting a specific protein that can be found only in the organism of interest. That makes sense? Let's say you have two samples. One sample supposedly contains virus. Another sample supposedly does not contain virus. We cool? So far, you follow me? You lyse the samples. You break them in pieces, specifically in proteins. So lyse them so they become a mixture of proteins. And then you put those samples, okay, on the arrangement that is called a gel. It is a gel. It looks like a gel. It walks like a gel. It walks like a gel. And you apply electrical field to it. Proteins have charge. They're going to be moving through the electrical field. Does that make sense? Which ones are going to be moving faster? Small or large proteins? Small proteins are going to be faster. So since they move, I'm not going into technicalities, since they move from up to bottom, the smaller will be at the bottom, okay? That makes sense because they move faster. So I separated them, separated proteins based on their size. Now what? Gel is incredibly inconvenient to work with. If you try to work with Jello, try to do something with the Jello, okay? So what we do next, we take a special material, special sheet that is called nitrocellulose. It's fine if you don't remember the name of the material. Again, I'm not going into technicalities. Pretty much you move proteins from the gel to that material. You put them together, again, apply electricity and proteins move to the material. Now you have a sheet with those same proteins. They are separated all the same way. Does that make sense? You have like an image. The difference is that these are actual proteins. And then you expose that membrane to the antibodies. Antibodies, in our case, we had a sample with the virus and without the virus. Right? That's what we started with. So imagine you have antibodies, already have, in your fridge, that can recognize virus protein. You add these antibodies and they bind very specifically to the virus protein. Does that make sense? They don't bind to anything else of that same size, only to this specific protein. Good? These antibodies have a way to get visualized. You end up with a picture where you have bands. Each band is a specific protein of a separate size. Does that make sense? Remember, I told you before, if it doesn't, I expect the answer, no, it does not. Do you have questions so far about it? Okay, so separate proteins and use antibodies to recognize a protein, to identify the protein of a specific size. This method is, I love doing Western blots because there's a lot of bench work, a lot of wet lab experience. Not that you're getting drenched, but you work with actual stuff. Like you do stuff with your hands, like cooking. Okay? You do stuff, and then you have machines. Like you feel like you're important in science. You feel sciencey. Okay. The downside it takes a lot of time. If you really like it, you start at 8 a.m., you finish at 8 p.m., but it has a lot of breaks. So you can pretend being busy and have lunch at the same time. Like you're sitting playing on the phone, what are you doing? Well, I run the Western blot. Well, kind of the whole thing goes on in the room for like an hour and a half, but you pretend to be busy, you know. You're doing something. Since they take a lot of time and it's really hard to make a quantitative analysis. It's more about presence. Western blots are not common in the clinical labs. The upside, they are extremely sensitive, like crazy sensitive. It's nano moles is absolutely not a problem. Like 1/1000th of a mole, it's easy. Okay. More common method is ELISA. You may have heard about it. It's not named in honor of a girl. It's actually an acronym. Enzyme-linked immune sorption assay. Again, it's a recognition of antigen by antibody. Initially, it was a direct ELISA. When you had antigen absorbed on the special plastic plate. You add antibodies. The more antigen you have absorbed on this plate, the more antibody will stick to it. Does that make sense? The more antigen you have, the more antibody going to stick to it. The more antibody is going to stick to it, the more signal you're going to have. Basic concept. Now, for detection of antigens, we mostly use sandwich ELISA. When you have antibodies that were pre-absorbed on the plate, you add your antigen, it binds, then you add other antibodies that produce signal and recognize the same antigen. For instance, if you're going to detect, well, I don't know, like, yeah, E. coli, H70157 strain of E. coli, it's antigen, you're going to do probably sandwich. ELISA, it's less sensitive than Western blot. The advantage is that it's simple, seriously. In terms of the hand skills, if you can hold a pipette and pipette, you're fine. Simple. It's really easy to teach how to do that. And it's easily automated. Essentially, now imagine you have a plate slightly bigger than the cell phone. It's a little bit wider and a little bit shorter. I worked personally with plates that have 96 wells. It sounds like a lot, but it's actually fairly easy to work. Then I was introduced to 384 wells, which sounded like crazy number, on the same surface. So wells become smaller and smaller. Now, there are plates that incorporate more than 1,500 wells. The only problem, you still have to manually put the samples on. So you have to be extremely accurate and precise. But then there are machines, you put your samples in the plate, you put it in the machine, and you wait for 2 or 3 hours. And you get the results. You don't do anything else. Okay, so it all comes down to sampling and result interpretation. So another advantage of that method, it's high throughput. Many samples in a short period of time. Immunofluorescence. Same principle. Antibodies bind to the antigen on the surface of the cell, or in some case, you can do it inside the cell. Then you look under the microscope and you see the pretty picture like this. This pretty picture was made by a friend of mine. So the red ones, neurons, the green ones are astrocytes, and blue ones are nuclei of the cells. So I have three colors in the same picture. Okay? Yes?
So, like, essentially with this method, you're just, like, throwing a bunch of antibodies onto a plate and seeing which one sticks to it?
Yeah. You talking about immunofluorescence?
Yeah, well, all of them, really.
Well,
Are you throwing
not
blood?
all of them involve plate, like Western blot does not.
You
But
want one specific.
you test antibody, and using antibody you test if there is an antigen. You look, did I answer? You just throw antibodies on whatever sample you have. The advantage of immunofluorescence, you can take a sample of patient's tissue, put it on like a slide, add antibodies, and see. And you can have like 3 different colors, so you can look at different things all at the same time. Does that make sense? Although, must say, this is absolutely not quantitative. So you can't say a lot or not a lot. That makes sense? Good? Any questions on immune? Yes. If you want to know the exact amount, yes. In many cases, you want to see if the patient has or has no, I don't know, HIV, whatever, plague, okay, cholera or whatnot. So you just want to know first if the patient has it. In many cases, it's not. Immunofluorescence is not quantitative in the terms that you can't say patient has 536,000 cells of staphylococcus per 1 milliliter of blood. Does that make sense? You can't say the number, but you can say, look, we have a sample of liver. It's just covered with amoeba, and there's nothing else. Or, and maybe a few specks of hepatitis C. Does that make sense? So you can still operate a lot and a little. Does that make sense? Biochemistry. Well, these 2 methods, they allow you to analyze biochemical features of the microbial cells. Technically, they are based on physics rather than the chemistry. And I know that when you say the word physics in class, half of the students just drop dead. 2 methods that we're going to talk about are chromatography and mass spectrometry. Chromatography. If I will take the Petri dish, pour some water into it, then take paper towel and dip it in the water, where would water go if it would anywhere? It'll go up, right? Capillary forces will pull the water up through the paper towel. What if I would add a little bit of watercolors to the Petri dish? Like, you know, the painting stuff, the artsy stuff. Like red, yellow, and blue. Yeah, they will go with the water. And if those dyes, the actual molecules, chemicals that are in the plate have different size, they would probably travel at different speed, right? So you will see bands, blue and yellow and red bands. That's chromatography. You can do it at home, actually. That's chromatography. Separation of chemicals when they go through some filter, some kind of absorbent material. I hope you're not going to argue that the paper towels are absorbent, right? And water travels through it. Of course, nobody uses this paper towel stuff in the clinic. What they use columns. Column is filled with some kind of absorbent material called stationary phase. And then you apply your sample. Sample consists of many components. You see 3 components here. And sample goes down through the material. Some components will travel faster than another. Does that make sense to you? Just the whole idea of them traveling at different speed. So the ones that travel faster will get out first. Well, second and third. Then it depends on what you want to do. You can collect fractions at different periods of time and analyze them. Or you can place some sort of a detector here that will check any parameter, like whatever you pick, parameter of this fluid that comes out of the column. Does that make sense? Do you think parameter, let's say it's going to be optical density. Will parameter change when one of those components come out? What do you think? Like, yeah, blue stuff comes out, it's probably different. Optical density is going to be different. Is there, can we think, is that conceivable that blue component will have different optical density than red one? Sure, yeah. So what you end up with is the graph like that, okay? Where each peak corresponds to a chemical, and the size of the peak corresponds to concentration of that chemical, amount of that chemical. Does that make sense? If you have controls, if you know that, I don't know, acetone comes out at certain time, then you can say this, based on the time, is acetone, this based on the time is this, that, blah, blah, blah. So it allows you in a pretty rapid fashion to analyze metabolites of the microbial cells. And you can imagine that cells may have like unique chemical signatures. The cell's going to produce a unique mixture of chemicals, and you can recognize that chemical signature of the cell. What will define the speed of movement? Which parameters will affect how fast the chemical goes through the column? Size. Absolutely, size. Something else. It's pretty much the same. Well, okay. Shape to an extent, but it corresponds with mass very well. Density, not really. It's dissolved. We talk about individual molecules. Charge, and the third one. There are 3 major ones. The chemicals. Composition, huh? Chemical nature. You're both right. Chemical nature. So that's why you can tell apart, say, acetone from formaldehyde. Because they're different. Chemically, slightly different. They may be similar in size, but they're slightly different chemically. Does that make sense? So it is a very powerful tool. The machine looks awesome. It's like a stack of sophisticated equipment, tubes running all over the place. It's fairly expensive, about $50,000 maybe. But you buy it once, and then all you need to change is columns. Pretty cool. Clinical labs use it. Next machine that we're going to talk about is even cooler. This one. British sci-fi writer Arthur Clarke. If you haven't heard, I strongly suggest you read it. Has a wonderful short story called 9 Billion Names of a God. Hilarious, grim, and short. One of my favorite sci-fi stories. Anyway, Clarke once said that highly advanced technology becomes indistinguishable from magic. And honestly, if you think about this, do we really know how it works? Like cell phone? I have like very, very basic understanding. Huh? Yeah, so a lot of stuff. I mean, my wife always says, when she looks at the plane, she thinks it's magic. She understands there's physics and stuff. But how this huge thing can fly in the air? So for me, this is the quintessentia of magic. Because this machine is a gigantic box. You put sample on one end and you get results on another end. And I know the basic concept, but what actually technically is going on there, I have no clue. So don't ask how it is done. I don't know, okay? So what is done? We have a mixture, a sample, which is a mixture of molecules. You put it in the ionization chamber. When chemicals are ionized, what happens to them? What is ion? Charged. So they gain charge. Charge may be positive, may be negative, right? They're going to gain different charge. Also, those molecules have different mass. And then, pretty much, this chamber shoots them out, accelerates them. Which ones are going to fly farther based on the size? Smaller, fly farther, bigger, land closer, right? If you place a magnet will change the trajectory based on charge. Does that make sense? You see, changing the trajectory. So the exact trajectory of each molecule, each ion, will be determined by mass and charge. Does that make sense? Terrific. What you end up seeing is the graph that shows the absorbance, well, the amount of a certain ion. And ion can be characterized by mass to charge ratio. A lot of fancy words. But the idea is when you put a specific, a mixture of chemicals in the machine, you get a unique graph. This is a signature. Essentially, when you take a sample of bacteria and put this bacteria in the machine, you get a unique signature, mass spec signature, that is characteristic only for this bacteria. And we even talk about strain, not even species. Does that make sense? So like if you put E. coli, it will produce one signature. You put Staph aureus, completely different signature. Then you compare it to the database. There's now a database of those mass spec signatures. And you diagnose your disease. It's absolutely awesome. Because, and we'll get to why it's awesome, many levels. Okay? So, the downside, you gotta have your bacteria in the pure culture. So you gotta have a sample. You have to grow it anyway. You have to grow it on the plate first, and then you can diagnose. Does that make sense? The whole process, from loading the sample to getting the results, takes about 2 hours, which is incredibly short. The biggest downside of this method is, any idea? $750,000 for a machine. Not every hospital, and hospitals aren't poor. Not every hospital, most of the hospitals can't afford it. Because you pay almost $1 million and you have to kind of justify expenses. Usually, you gotta have a huge flow of samples to justify expenses. If you analyze one sample a day, it's probably not brilliant idea, you know, to bring this in. That makes sense? That's really awesome. I love this whole idea. Now, one thing that I want to mention, going back for a second, when we talked about immune-based assays, when you take antibody to test, which antibody you take? It's simple. When you're about to test your sample, which antibody you take? How you decide which antibody to take? I mean, I have a sample of blood. The patient has sepsis. How do I decide? Which antibody I take? It's not like entirely random, but it's kind of my deliberate decision. So if I think it's E. coli, I'm going to run for E. coli. What if it's not? Will I know what it is? Like, I test for E. coli, it comes out negative. Do I know what it is? No. So you find what you look for. Does that make sense? In a lot of these methods, you can find only what you look for. You know, like when you are in the crowd and you look for someone in that crowd, you can miss a bunch of people that you already know because you don't set your mind to recognize them. Does that make sense? Same here. That's important. This method allows you to pretty much see. You don't know what the sample is, but you're not restricted. You run it, you check it. You have 5 different kinds of colonies, you can check all of them. Does that make sense? So it doesn't have this restriction. Last, genetic methods. Polymerase chain reaction, who've heard about it? PCR. Okay. So it's a pretty neat method. The guy who Kerry Mullis, I recalled his last name. Everybody says he's an asshole. Seriously, like Kerry Mullis, the guy who developed the PCR, scientist, PCR, polymerase chain reaction, right here. He and his colleague got Nobel Prize for it, but I've heard a lot of like phrases that he's quite a character, which in scientific world means he's pretty much an asshole. On the personal level. Anyway, DNA can be copied. When I make a statement like that, you are allowed, absolutely, you're welcome to disagree with me and ask, really? And I will explain. But do we agree that it can be copied? There are cellular enzymes that copy DNA. Okay. So Mullis and whoever the second dude was, they decided to use it to amplify the DNA molecule, to create new exact copies of DNA molecule in the test tube. That may sound absolutely useless unless you put some restrictions. DNAs of different organisms are different, right? DNA of virus, whatever virus, influenza, will be different from a DNA of RSV, which is another virus. That makes sense? Well, they're not DNA viruses, so it's a bad example. Let's say DNA of Streptococcus is going to be different from DNA of Staphylococcus. Do you agree? Do you agree with me? Right? So not completely different, but there are going to be fragments that are very, very, very much different. That makes sense? So Mullis and that other dude decided to develop a method that would allow you to in a very specific fashion replicate and accumulate the fragment that you want, fragment of a DNA. Does that make sense so far? So what they've done, what the method is about. So I have a DNA, which is a double strand. In this case, the bases A, T, G, and C interact with each other by hydrogen bonds and cannot really be read. And that's what we want to know. You know, that's how we know that it's specific, because of the sequence of the bases. Does that make sense so far? How can you break down hydrogen bonds? Hydrogen bonds, like the ones that hold water molecules together. Huh? Heat, yes. You heat it up. So you pretty much boil your DNA. DNA is tough, it's fine. Strands get separated. Now your bases, the nitrogenous bases, are open. So you can start kind of reading them, recognize them, right? The first thing you do, you add special small DNA fragments called primers. Primers are designed so they bind to a very particular part of DNA. And they design in a way that primers are absolutely unique for the species that you're looking for. Does that make sense? So you can make unique primers that will bind to some parts of a gene that can be found only in Streptococcus pyogenes. Only, not any other organism on Earth. Does that make sense? They're absolutely unique. These primers, as you can see, they set some sort of a starting point for the enzyme known as DNA polymerase. DNA polymerase gets here at the place where primer is complementary bound to the original DNA strand, and it starts to copy the old strand, making the new one. That blue is the new one. That makes sense? So DNA polymerase has the restriction in terms of the starting point. Does it have the end point on that during that step? Does it have the end point? No, it will go as far as it can, right? And then it will eventually follow. But what's going to happen next? What can you say about these two new strands? How they relate to each other in terms of binding? Are they complementary or not? They are, right? So these strands will then bind to each other. And this time, you do have a restriction, because this end ends here, right? This strand ends here. So eventually, you're going to start accumulating short fragments of DNA that are restricted by the primers you have selected in the first place. Remember our analogy of a cookbook. Imagine that somebody tells you, copy the fragment of your cookbook that starts with the word bucket and ends with the word snowman. Don't ask me what snowman does in the cookbook, but anyway. To find the, and I bet that, huh? Okay, well, imagine that you have only one word bucket and only one word snowman in the whole cookbook. So there's going to be a unique sequence restricted, unique fragment of the cookbook restricted by those two words. And you're going to copy only what's between them. Does that make sense? That's what PCR does. Now, these fragments, as we discussed, they are absolutely unique, and you can visualize them. You can look at them. You can separate them. You can look at them under the gel. There are many methods. So far, does that make sense? Now, if you have two samples, say we have primers against Abna protein of Epstein-Barr virus, whatever. So we have two samples. We run PCR on both. We see these fragments in one, and we don't see them in another. What does that mean? That means that in the sample in which we see fragments has the virus, has that gene, has that DNA fragment that we were looking for, meaning that there's a virus. The sample that's negative means that there's no virus. Does that make sense? When you donate blood, they test all blood samples for HIV, all of them, no exceptions. They do it by PCR because, again, this method can be easily automated and it's high throughput. So the only thing that you have to do, well, actually there are machines now that you just load the blood sample in, they will isolate DNA, transfer it to the PCR machine, and you get the actual result, like positive, negative. Some machines will even tell you how much DNA, how many molecules you had in the initial sample. Yes? Question? No? Okay. So this is a pretty neat method. Why I'm so excited about it. Every time, every cycle, so this is going to be like one cycle. At the end of first cycle, if you start with one molecule of DNA, how many molecules you have at the end? 2. At the end of a second, at the end of a third, 8. At the end of a 40th, F40, I'm not asking for exact number, I'm going to 10 in the power of 12, and a change. A lot. 40 cycles of PCR, if you do it like, not manually, but like if you prepare it like in the lab, in a research lab, stick it in the machine, it's about 2.5 hours. It's incredibly fast. Gives you very quick results. It's one of the most common, most popular methods for sample analysis, and it's very sensitive. We now have specific techniques that, technically speaking, let us to detect one molecule of DNA in the sample. That's mind-boggling. So, I mean, it means that, you know, when you look at the CSI, when they collect like cigarette butts and somebody drinks from the coffee cup and then they swap, that is true. When you drink, you leave your genetic footprint all over the place. Trust me, it's everywhere. If anybody would want to find it, they would find it. The culmination, the climax of this whole story. I feel like a televangelist at some point. But I love that. Sequencing. This is, with mass spec, I have a vague idea of what's going on inside. With sequencing, I have absolutely no. I don't know what's going on. I mean, I know what uses a sample, and I know what kind of result it gives you. What happens in between, listen, it's like magic for me, seriously. So what it does, well, it's going to be easy. It determines the sequence of nucleotides in the DNA. So it pretty much reads the genome. Does that make sense? Why is that so exciting? Yes, well, you probably won't be able to say if your child's going to be playing piano, okay, or will be extremely talented in art, but it will be able to tell you if the child has any genetic deficiencies that are not apparent in the first place. Do we have overall, like, general human genome sequenced? Yeah, we do. It took something like 10 years or something, and it cost us $200 million to sequence one genome, like a human genome. 2 weeks ago, well, roughly, the company named Illumina, they make biomedical devices, announced that it's going to release a new device on the market that will allow to sequence human genome in several hours for $100. Which means you walk into the doctor, you take a blood sample, they'll call you next week, tell you, dude, you have 4 genes, 4 copies of a gene that are responsible for resistance to alcohol, so you can drink as much as you want. Okay? Which is a future of medicine. We're going to deal with the genetic makeup of organisms. Now, this is all pretty awesome. How is that applied to microbiology? Imagine that you have fecal sample. You can take fecal sample, run it through sequencing machine, compare it to databases of known genomes, and know exactly which microbes you have. That makes sense? What's the problem with that method in terms of clinical diagnostics? Yeah, you get overwhelmed, right? Have a patient with diarrhea, and you get 1,500 species from the stool. It's overwhelming. You don't know which one to blame, right? So, for directly diagnostic purposes, this method is rarely used. But one of the amazing applications of sequencing that I know of was its actual clinical case. Since I have a horrible memory for details, I won't be able to tell you the exact age of the girl. She was really little, like maybe 6 months or 2 years, whatever, doesn't matter. Little girl comes down with the flu. Not just flu, she's about to die. She's doing very, very bad. Other than that, all the time, she was healthy. She didn't show anything, I mean, nothing, everything was fine. Phenotypically, she was absolutely healthy kid. And out of nowhere, well, she had like diarrhea from time to time, maybe, the cold, and flu just knocks her down. Doctors, since they were puzzled what's going on, they sequenced her genome. Now, before Illumina announcement, it cost about like $1,500. I mean, it's affordable. You can do that. They sequence the genome, they look at the genome. Turns out she misses one of the key proteins in the innate immune response. When she's healthy, it's not reflected. Not even every pathogen will kind of require that protein. But influenza, the protection against influenza was tied to production of that protein. She didn't have the protein, she came down really sick. So they now at least knew what to play around, what to start with. And they managed to treat her, she survived, but now she knows that she has to be aware about certain types of diseases. Does that make sense? It's really, really cool. The sequencing is widely used for epidemiology. If you want to see the distribution of microbes, you probably have heard that people who eat high carb versus like low carb diet have different intestinal microbes. It was all done by sequencing. We're going to pause.